We're here today at the labs at the Feline Health Center as part of Cornell's College of Veterinary Medicine, which is where we're developing this new surveillance analysis of feline coronaviruses, which has really never been done before. We have a lot of information on many coronaviruses, including the one causing the COVID pandemic, but we have very little information on feline coronaviruses, which is something we're trying to address with new sequencing techniques, but also to transform that into a new test or a risk prediction for FIP, because the conventional tests really don't discriminate the widely circulating benign virus from the lethal pathogenic FIP virus.
Unfortunately, we don't have any good antemortem testing options for FIP, particularly for the cats that are not showing effusion. There are a number of diagnostics that can be used as an aid to the clinician. However, the antibodies produced by FIP and by feline coronavirus, which are actually the same species, are indistinguishable. And so just having a titer for feline coronavirus is not confirmatory for FIP. In studying samples from cats of feces and blood and various tissue biopsies, we have been trying to tackle something that's been very elusive for feline coronavirus and all the alpha coronaviruses. There's so much variation that it's technically very challenging to get large portions of the spike gene sequence. And so we are working with several types of new sequencing technologies that are going to enable us to not only cover the full length of the spike gene and perhaps the entire genome, but also do so at a very deep level in order to be able to catch that variation when it happens.
And there's one particular area that we've really focused on called the furin cleavage site. This is actually also present in SARS-CoV-2, the cause of the COVID pandemic. So that furin cleavage site is really a powerful way for a virus to alter its properties. It can increase transmission, it can induce changes in cell tropism. So the goal right now is to use the obvious changes in that furin cleavage site, which we could identify in an FIP cat, but also think about what's happening prior to that. What are the background changes? It would be called epistasis is the word in genetics. It's not just one mutation, it's a viral evolution that's occurring within a given cat, and we're trying to track that over time to really understand how the virus is evolving.
The technology is able to sequence the same region of the virus's genome many times over, many thousands of times over, and so we can look wide scale at the population, but then we can also look at the individual cat and look at how the virus is mutating within an individual cat or not.
So we use two technologies. One that is this small platform, that's the nanopore, that gives us really fast sequences, and another one that is not so fast, but it gives us a lot of depth and a lot of sequences. We do a multiplex PCR in which we target several different genes of feline coronavirus. We run them in a single PCR and then we sequence them all at the same time. So you just load the sample, and then in about an hour we have gotten around 100,000 sequences of the 3 targets that we have so far, which is really fast and really tells us a lot about feline coronavirus.
The goal we have with our deep sequencing technology is to be able to separate out the two forms, ideally from a fecal sample or perhaps a blood sample as well. That will be a huge aid in terms of screening healthy cats and management of sick cats and being able to isolate them before the disease is completely evident.
New antiviral drugs. They're being used quite extensively in clinical practice right now and apparently working incredibly well. They're a cure, if not a solution, to the FIP problem, but we need to be good stewards of these drugs. We need to make sure that they're used at the right time in the right cats with the right dose to avoid any side effects, to avoid cost to the owners. There's lots of factors that go into this. We need this basis of risk assessment behind the scenes to really understand how to manage those drugs. We have a population of viruses in the cats. It's not converting from one virus to another. It's a process of conversion from FECV to FIPV. So a given cat may be shedding both forms of the virus at the same time. And it may be that we've got a very sick cat, which is actually shedding the so-called FECV form at a higher level. So that's where conventional PCRs aren't helpful, because we're actually picking up the wrong virus. So this is where we need to go and look at that population.
And we've been really fortunate that Feline Health Center members have donated samples from their sick cats to this research program, which we have been working on also with the generous support of Feline Health Center donors. And so now we're really taking a look at all the samples that have been sent to us and getting that information about the variation over the course of the disease and within the cat in different tissues. And we're hoping to be able to put all of that together into a risk assessment tool that will be accessible to everyone within the next couple of years with continued support.
If you'd like to join us in our mission, please consider making a donation or becoming a member of the Cornell Feline Health Center.